Hello and welcome. Today, I am going to discuss with you a very important aspect in molecular genetics, that is genetic analysis. The genetic analysis which I am going to describe it involves the genetic variation or variability obtained and observed by way of mutations in a population. So you have a wild type population and you have a mutant population. And then combination, the generation of newer combinations. Okay. Now, this kind of analysis has given wealth of knowledge, basic concepts, all came by doing simple genetic analysis. Okay. Like, see, the gene mapping was done. The gene order on a chromosome was done by using this kind of genetic analysis, which I'm going to describe and discuss with you today. And also, we could know how many genes are involved in a metabolic process in bacterial systems. And also, we could differentiate between regulatory site and regulatory genes. It also gave rise to a concept of operons in prokaryotes or in bacteria. That is, all the genes involved in a metabolic process are located in a sequence together and are expressed or not expressed at the same time. And they were also under the regulatory control of a single switch. So all these kind of concepts came by simple genetic analysis. And today I'm going to discuss with you what this analysis was at that point of time and is even today. It is the approach is being used in basic microbiology and molecular genetics. So when we look at the what are the tools which were available and utilized for genetic analysis? So there are basically two processes, or you can say tools, that is genetic recombination and complementation. Now, genetic recombination is the key for genetic variability, what we see today in eukaryotic world as well as prokaryotic world. I'm going to focus on prokaryotes, not on eukaryotes. So here also, recombination or the genetic recombination is the key to have the genetic variability seen in a population. You see different combinations arising out of in a single population when a cell divides, or by way of presence of an extra allele in the cell. So when it divides, it undergoes recombination. Recombination means it generates newer combinations. Okay. Similarly, when we look at the complementation, complementation again is the key where one sees that if there are two alleles, and if out of these two, one is defective or not expressing, and another is expressing and is complete, is not defective, then how this extra copy or extra allele complements the defective one, and it results into regaining the wild type phenotype. So there is a change in the phenotype after complementation. And how the complementation is done, that is by way of having an extra allele, what we call an extra copy of the same gene, which is defective either here or there. So in this kind of scenario, it tries to complement. So today I'm going to discuss one of these tools, that is genetic recombination. We will understand in more detail that what this genetic recombination is and what kind of information one derives from this analysis, which uses genetic recombination as the tool. Okay, so let us look at this cartoon. Now here I have tried to show you here that how this recombination takes place. So if you have uh, two phage DNA and one of the phage, parental phage, is defective in a trait which I am referring as B minus or A or B. Okay, these are the two traits you can take that. So one is one parent is A plus and B minus. It means it shows the A trait and doesn't show the B trait. Okay, so A plus B minus, and the another phage is A minus and B plus. That is, it is defective in A trait and it is normal for the B trait. So now, if these two phage DNAs are present in the same cell, then what happens? There is exchange of segments at this point. You know, so as a result, what you see that this B plus moves and sits closer to A plus. Okay, so it is the breaking of the chromosome or the DNA here and resealing takes place. So as a consequence, when you look at the population, you see a wild type and a double mutant, because this B minus has moved from here to here. So it becomes a double mutant, that is A minus B minus, and it becomes a wild type, that is A plus B plus. So that's how the recombination takes place. So recombination or the movement of DNA fragment from one to another results into a newer combination. Now, if you look at the parental type, the parental is A plus B minus or A minus B plus, but after recombination, a new combination was generated, that which was A plus B plus, and which is the wild type. Okay, so this is what we mean by recombination, where physically the DNA segment is broken at one point or cut at one point and sealed or ligated in today's term or joined with the other segment to give you a changed phenotype. Okay, so it's a newer combination, and that is what in eukaryotes also. I'm not going to refer there. The lot of newer combination in each generation after as the one undergoes sexual reproduction, you see a newer combinations coming. Forget that. So now, so this phenomenon has been exploited for deriving a lot of information about bacterial genome in 60s. Okay, so further, if we try to understand that how genetic recombination has helped in gene mapping. Okay, so this process of recombination or event of recombination, when it takes place, it happens at a frequency. Okay, at a frequency, that is, in a population, not all phages will show the recombination process. Okay, so basically, we try to look for a frequency at which this process is occurring, and how that frequency is decided or determined. That is by taking number of recombinant phages or recombinants. It can be bacteria, it can be phage. So we look for number of recombinants divided by total number of individuals, whether phage or bacterial cells. Okay, so that is to decide the frequency at which the recombination process is taking place. Okay, now this recombination process tells us a few things. That is, by looking at relative frequency or the recombination frequency, we can know what is the distance between the two genes. Okay. Okay. So how close or far those two genes which we are considering are located on the on the DNA. Okay. And also, if we look at the linkage process uh, between the two genes, we can also know the position or the gene order, for example. Okay, so recombination frequency, I mean, which came from process of recombination or event of recombination, where physically DNA is moving from one DNA to another. Okay, so how frequent that process is or event is, that tells us two things. One is that how close and or distant the two genes which we are considering are, and also whether they are linked or not. Okay. Now, further, if we look at as an example, okay. So, if see, this is hypothetical. If you have the two phages, this is one is donor, another is recipient, and if you see, one of them is A plus, B minus, C minus, and another is A minus, B plus, C plus. Okay. This is donor and this is recipient or donor, whatever way you want to take. Now, if there is a cross between these two or recombination between these two, okay, you can get newer combinations generated. Okay. Now, before we proceed, you can see one more thing here. If you notice here that the distance between A and B and C is equal. They are equally spaced. Okay. The distance between A and B and C is same. Okay. Equidistance. Now, and if you see the distance between A and C, that is twice the distance between A and B. Okay, it's as simple as that. See, if this is 2 centimeter, this is 2 centimeter. Okay, but A and C would be 4 centimeter. C is away from A by 4 centimeter as an example I'm trying to give. Okay, now the possibility of recombination between A and C, okay, and A and B. So if you look at the recombination possibilities between A and B and A and C, just by logic, you know, the A and C recombinants will be more as compared to A and B. Why? Because the distance between A and C is more than A and B. And therefore, you get twice the number of A plus C plus as compared to A plus B plus recombinants. Okay. So a general law or conclusion or rule was drawn that recombination frequency is directly proportional to the distance between the two genes. The recombination frequency is directly proportional to the distance, between the two genes. Okay, and therefore one can determine the relative position between the two genes. For example, in this case, if you see, we can say that B is sitting closer to A as compared to C. C is far away from A. Okay. Now, further, if we see as an example, if in an experiment you see a recombination frequency between A and B is 1% and B and C is 2%. Okay. Okay. What does this mean? This means there can be two gene orders. Okay. One can be this A, B, and C, and another can be C, A, and B. And in these experiments of this kind, we first put an hypothesis. If we are looking for an gene order between A, B, and C, so and look for recombination frequency between A and B and C. Okay, so here there are two possible gene orders. One is ABC or CAB. There cannot be third order. presumed. Okay. Now, if you look at A and B recombination frequency is 1% and B and C is 2%. Okay. Now, if in the second gene order, C, A and B, so A and B is 1%, and B and C is if the A and C is 1%, then the initial B and C frequency, recombination frequency is 2%. So 1% plus 1%, this is 2%. So considering these two, this data of recombination frequency between A and B, B and C, you see these either the order can be ABC or the order can be CAB. We cannot decide until and unless we also know a third recombination frequency between A and C. This is very important. If you know what is the recombination frequency between A and C, then we can decide that which one of the two gene orders exist. How? For example, if the gene, the frequency between A and C is 1%. If the frequency or recombination frequency between A and C is 1%, then this is the gene order. And if it is 3%, the A and C recombination frequency is 3%, that is 2 plus 1, 3%, then this is the gene order. So just by looking at recombination frequency, we can know a few things very clearly, that what is the relative distance between the genes and what is the gene order by doing this kind of analysis. Now, if we add or consider another factor into it, that is linkage. Now, linkage is a process which is from eukaryotes, and of course, all these things which I'm talking about has a root in eukaryotic genetics. So, linkage. Now, in eukaryotes, if you remember, all the genes located on a chromosome are linked. Why? Because they cannot undergo recombination. The recombination in eukaryote takes place between the genes located on two different chromosomes. Generally, okay? So this concept that all the genes located on a same chromosome are linked and they are not able to undergo recombination, okay? So similarly, in prokaryotes, if we look at, in prokaryotes, you have only single circular chromosome. It means you have only single linkage group. In eukaryotes, you have as many linkage groups as the number of chromosome an organism has. So in prokaryotes, since there is a single circular chromosome, you have only single linkage group. Okay. So in this, if the two genes are located close to each other, or they are so close that no recombination is possible, then we call them as linked genes, or the genes are linked. Means the two genes which are not able to undergo recombination on the same chromosome, we call them as linked. And if the two genes are far away from each other and can undergo recombination, they are, they are not linked or unlinked. So this concept also helped in further analysis and try to find out what are the genes which are linked to each other and what are the genes which are not linked. So how this analysis is done, let us look at it as an example. For example, here. Now, if we consider a cross between two parents, where we have where you have the parental genotype A plus B minus C plus, or A minus B plus C minus, that is A plus B minus C plus, and the second parent is A minus B plus C minus. Okay, so these are the parental. This is the same, A plus C plus, only the order is different. Here A, B, C, and this is A, C, B. Okay, and this is the another A minus C minus B plus A, C, B or A, B, C. So again, as I mentioned, that you have to first put a hypothesis in such experimentations, that okay, these are the two things possible, and when you get the data at the end of the experiment, this data will either support uh, the order 1 or the order 2. So okay, now let us look at this, what is happening here. So you have uh, if there is a cross between these two parents, okay, which is A plus B minus C plus or A minus B plus C plus, and if we look for B plus C plus recombinant, B plus C plus recombinants, and ask a question whether that B plus C plus is A plus or A minus. Okay, so that is the experimentation that we are allowing them to undergo cross or recombination, and we look for B plus C plus phenotype, and those cells which are B plus C plus, we ask another question and check those cells whether they are A plus or A minus. Simple. Now, if you look at these arrangements also, if you see here that A, B and C are sitting close to each other, okay? And A is far apart. Similarly here, this is the replica of that basically. So what we are trying to see is that B and C are closer to each other as compared to A, and B and A or C. Okay, so when we get this B plus C plus, or we get that A plus B plus C plus phenotype, and similarly here you get A plus C plus and B plus. Okay, I mean both B plus C plus are also there A plus. So you get a recombinant wild type phenotype and a triple mutant basically. Okay, now the question is, what is then gene order, whether it is ABC or it is ACB? Okay, now this question is answered by looking at recombination frequency. Okay, that is, out of the two, which one is more frequent? That is, see, if you have this scenario, A plus and B minus C plus, to get this B plus, this has to be recombined. And this recombination is involving two exchange or two crossovers, one at this point and one at this point. Then only this B plus will move here and B minus will move here. Okay. But if you look at this panel here, the A plus, C plus, and B minus, but this B, to make it B plus, there has to be a single crossover taking place between B and C or C and B. Okay. So to get A plus, C plus, B plus. Okay. A plus, B plus, C plus. Okay. So in these two panels, one recombination is requiring two crossover processes, and the other is requiring only single crossover. And it is known that frequency of two crossover is less than single crossover. So in an experiment, if you see the recovery of A plus C plus B plus is more, then this is the order. And if it is less, then this is the order. Because the order ABC will require two crossover, which occurs at a lesser frequency. And therefore, if you are observing a very less recombination frequency, then this is the panel 1 is the gene order. But if you are getting high frequency of recombination and recovery of A plus B plus C plus, it means it is disorder because it is involving only single crossover. Okay, so just by looking at the recombination process, you can give the gene order. Okay, let us look at another scenario wherein we can perform less crosses between the two and derive a very useful information here. Uh, for example, um, if you look at here, you have again, like this, earlier we looked at A plus B minus C minus one donor and the recipient, or recipient and donor A minus B plus C plus, and A plus B minus C minus and A minus B plus C plus. Now in the, see again, these are the three traits we are looking at. ABC are nothing but the traits. You can have anything replacing ABC. But what we are saying, we are considering one scenario where a cell is plus for one phenotype and minus for another, or mutant for another. Minus means mutant for another two phenotypes. Okay, and the donor is a mutant for the A plus, and it is the wild type phenotype for B plus and C plus. Okay, now in the two panel, if you see, in the two panel, if you can see the difference, in this, the distance between A and B is more as compared to B and C. But if you look at the panel 2, the distance between A and B is less than B and C. Okay, so what we are trying to show here, that in one panel, we are considering that B and C are linked, and in panel 2, we are considering A and B are linked. Okay, so when you cross these two, um, and then look for A plus C plus phenotype, A plus C plus phenotype, and ask a question that out of this A plus C plus, how many are B plus? Okay, so uh, how the A plus and C plus will be obtained? If there is a recombination taking place between A and B here and B and C here, because the gap between A and B and C is more. So possibility of a recombination is here more and here more. Okay. So if if you are getting A plus B plus A plus C plus phenotype and asking a question whether it is B plus or B minus. Okay. Now, if 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 your A plus C plus is also B plus, then what it means? It means this is the gene order. That is A, B, and C. Because if there is a recombination or crossover at this point, you get A plus, B plus, C plus. Okay. Similarly, if we are looking for A plus and C plus phenotype, but if it is B minus, okay, it means the crossover is taking place here. You see A plus, B minus, and C plus. So in just one crossover, you can decide the gene, the relationship between between A, B, and C. That is whether A is closer to B or B is closer to C. Okay, like whether this is the arrangement where B and C are closer, or this is the arrangement where A and B are closer. Okay, so it's a simple analysis. Okay, so by doing this kind of analysis, where genetic recombination process was taken as a tool, and lot of information was generated to understand basic bacterial genetics in 60s. Okay, so today I will stop here, and in my next video I will discuss how the another event, that is the complementation process, has helped in understanding uh, very basic aspects of molecular genetics, the organization of gene into operons, and how the regulatory and the sites and the regulatory genes were discovered. All that we will understand in my next video, which will be released very soon. So thank you very much for patient hearing. If you have liked this video, please subscribe my channel and hit on the bell icon so that you get notification for my subsequent new videos. Thanks again.